In May of 2026, a research team from Curtin University lowered a water sampling device into a submarine canyon off the Ningaloo coast of Western Australia and pulled it back with something that had no business being there. Not a body, not a carcass, not a beak washed in from open ocean. DNA, living DNA, shed hours earlier by an animal the size of a school bus, in a location where that animal had not been scientifically documented in over 25 years. The animal is Architeuthis ducks, the giant squid, the largest invertebrate on Earth, 13 meters long, carrying eyes the diameter of dinner plates, the largest eyes of any living creature. An animal that until 2004 had never once been photographed alive in its natural environment. Its presence off Western Australia was not supposed to be possible. The last confirmed record from that coastline was from 1999. A single-stranded carcass that told scientists nothing about whether a living population existed nearby, or whether it had simply drifted there from somewhere else. The 2026 signal was not a carcass. It was a live presence confirmed by genetic traces, shed skin cells, mucus particles, biological waste that degrade within 48 to 72 hours in seawater. Whatever left those traces was there alive within 3 days of the sample being collected. Why there? Why now? And why, after 25 years of scientific silence off that coastline, did something the size of a city bus suddenly appear in the dark below Western Australia? Before you can understand what this detection means, you have to understand what it does not mean, because the first instinct the internet ran with was wrong. The giant squid did not surface. The eDNA was collected at a depth of up to 4,500 meters deeper than the wreck of the Titanic, deeper than most military submarines are rated to operate. Rule that out immediately. This was not a surface encounter. What about geographic range? The Ningaloo coast faces the Indian Ocean. Giant squid have been documented in the South Atlantic, the North Pacific, Antarctic waters. The landmark 2004 footage by Tsunemi Kubodera of the National Science Museum in Tokyo was shot off the Ogasawara Islands. 9,000 kilometers from where this eDNA was found, Western Australia was considered peripheral. The last scientific record before 2026 was 27 years prior. A natural drift does not explain it. A carcass can float thousands of kilometers, but living eDNA degrades rapidly in warmer surface waters and breaks down over time. Whatever the Curtin team found in that canyon was local, recent, and should not have been there. The study was led by Dr. Georgia Nestor, a researcher at Curtin University and the Western Australian Museum, with results published in the journal Environmental DNA. The method was environmental DNA sampling, eDNA, which allows researchers to confirm a species' presence without ever directly observing it. Every living animal continuously sheds skin cells, mucus, and biological material into its environment. In cold, deep, low UV canyon environments, this material persists long enough to be collected and sequenced. The team deployed samplers across the Cape Range and Cloates submarine canyons. They were not looking for giant squid. This was a broad biodiversity assessment of the canyon system. The Architeuthis signal was not expected. When sequencing came back, they ran it 3 times. It matched, all 3 times. Here is where the case becomes genuinely difficult to explain. The Ningaloo canyons are not typical giant squid habitat. Everything the science tells us, from sperm whale stomach surveys to a century of coastal stranding records, points to a preference for cold, deep mesopelagic zones in temperate to subpolar latitudes. The Indian Ocean off northwestern Australia is warm. Surface temperature at Ningaloo in May 2026 was approximately 26 degrees Celsius. Giant squid blood uses a copper-based protein called haemocyanin to carry oxygen, a system that becomes dramatically less efficient as temperature rises. In warmer water, these animals cannot sustain the oxygen delivery their muscle tissue requires. They are not supposed to be in that ocean, and yet the DNA confirmed they were. To understand why, look at what has been happening in the Indian Ocean. Research from CSIRO and international oceanographers shows that the Indian Ocean has been warming significantly faster than the global ocean average over recent decades. The deep water upwelling systems along the Western Australian coast have been disrupted. At the same time, Deep sea monitoring data shows that prey species which historically occupied shallower depths have been shifting deeper into the canyon floors to remain within cooler thermal niches. The prey move down. If you are a giant invertebrate following food, and the food drops deeper into a canyon system your species has not historically occupied, you follow. The detection does not prove a permanent population or active breeding. What it proves is that in 2026, giant squid were present in waters they were not supposed to inhabit, while every ecological system around them was under measurable stress. Two ways to read that. One is optimistic. The species is more adaptable than we thought. The second is not optimistic at all. But here is the part the paper itself does not resolve, and it changes what the detection actually means. eDNA tells you an animal was there. It cannot tell you whether this was a natural range expansion or the signal of a population already in serious trouble. Stay with us. Giant squid have no established population count, no global census exists. These animals live at depths where direct observation remains nearly impossible even with current submersible technology. What we have instead is proxy data from their primary predator, the sperm whale, satellite telemetry and passive acoustic tracking of sperm whales in the Southern Ocean and Indian Ocean show these deep diving mammals spending longer underwater and foraging at greater depths to locate food. The inference is uncomfortable but consistent. Their prey is becoming harder to find at traditional depths. If the giant squid population were healthy and well distributed, eDNA signals would appear broadly across multiple canyon systems, multiple depth ranges, multiple independent detections. Dr. Nestor's study documented the signal in one specific canyon system at extreme depth, in a region with no prior confirmed record in over 25 years. That is not the fingerprint of a thriving population expanding confidently into new territory. That is the fingerprint of animals pushed to the margins of a range that can no longer support them where they used to be. The study language is precise and measured, but read between those lines and the implication is clear. The giant squid did not appear in the Ningaloo canyons because conditions were ideal. It appeared because conditions elsewhere had already changed. The canary in the coal mine is sometimes the last thing you see before the air gives out. The giant squid is not the top of the food chain. It is the pivot point in the middle of it, an apex predator of deep ocean mesopelagic zones and simultaneously a key prey source for the sperm whale. A single adult sperm whale consumes up to 1,000 kilograms of food per day, with deep sea squid forming the vast majority of that intake. The global sperm whale population, estimated between 300,000 and 800,000 individuals and classified as vulnerable by the IUCN, relies on these deep ocean food webs. If giant squid are forced into marginal habitats, the effect does not stay contained to a single canyon, it cascades. Sperm whale populations dependent on predictable prey grounds face increased foraging strain. Deep water fish populations shift unchecked. The vast, invisible machinery of the deep ocean begins to move in ways current ecological models are still trying to understand. We cannot count giant squid directly. The ocean is too large and the animal lives too deep. What eDNA sampling has done, for the first time in marine biology history, is give us a window into a part of the ocean that was previously dark to science. And the first clear look through that window off Western Australia in 2026 did not show us a species thriving in a new frontier. It showed us an animal in a place it was never supposed to be. Research teams plan to expand eDNA surveys to additional canyon systems along the Western Australian coastline in upcoming expeditions. If they find more signals across a broader footprint, a clearer picture emerges. A population, a range expansion, a species adapting. If they find nothing further, if the 2026 detection remains an isolated signal, then the question with no comfortable answer becomes unavoidable. What happens to deep diving predators when their primary prey can no longer be found where they expect it? And what does it mean that we only knew the giant squid was there at all because we now have technology sensitive enough to detect what an animal left behind in the water, not the animal itself? The ocean hides things science has not yet learned to look for. The 2026 detection is proof of that. Tell me in the comments, is this evidence of a species adapting and expanding, or the first readable signal of a population already retreating? The scientists who found it are still investigating. That argument belongs in the comments.